Hi, welcome to the Bioinformatics Chat. Today I'm speaking with Linus Schumacher. Hi, Linus.
Hello.
So, Linus, you wrote a couple of interesting papers recently. One of them is multidisciplinary approaches to understanding collective cell migration in developmental biology, published in Open Biology, I think, last year. And the other one is semblance of heterogeneity in collective cell migration, published this year in Cell Systems. And I want to talk to you about multidisciplinary approaches and about cell migrations. That sounds like it's a lot of fun. Before that, tell us a bit about your background.
Yeah, so my background has been quite interdisciplinary. When I finished high school, I was torn between going to university to study physics and going to study biology. And one option that enabled me to sort of delay that decision was to study a course called Natural Sciences, in which I mostly studied physics, but could take a lot of other subjects such as physiology or material science and neurobiology. And I continued that for my undergraduate, specializing in sort of a core physics education, but always taking the opportunity to work on biological problems, and pretty much continued that throughout my PhD as well, which was again an interdisciplinary training program.
Where did you do your PhD?
So my PhD was done at the University of Oxford and working mostly in the Centre for Mathematical Biology, which is a group of mostly mathematicians, but a lot of sort of computer scientists and physicists as well, doing theoretical work in application to biology.
Right. So this problem of cell migration, was it the first problem to which you tried to apply this multidisciplinary approach?
There were a couple of other problems along the line. I was quite fortunate to have the opportunity to do some short research projects before settling on my PhD. It was sort of, one of these doctoral training centers they have in the UK, which are probably inspired a little bit by the US PhD, in which you have rotation projects. So I first worked on some problems in auditory neuroscience and in stochastic pattern formation, which sort of introduced me to the world of mathematical biology, and I really enjoyed the group and the environment there, but at the same time wanted to work in projects that had a close collaboration with experimental biologists. And for that reason, I chose the cell migration problem with applications to developmental biology, because that's just a fascinating area of biology that's ripe with problems where we can make quantitative contributions. And that's one of the things I try to outline in this sort of perspective article in Open Biology last year.
Mm-hmm. And so when you first came across cell migration, was it already a problem to which people were trying to apply multidisciplinary, like mathematical and physical approaches, or was it your idea?
No, I can't claim that it was my idea. The history of computational and mathematical biology is quite rich, and the problem of cell migration has not escaped that, of course. I really joined a collaboration that was already ongoing with a great collaborator, Paul Kalisa, from the Stowers Institute of Medical Research in Kansas City, who did a PhD in mathematical biology himself and then transitioned to doing his own experiments to test the predictions of his mathematics, but then being left with sort of too much stuff to do, which is why he kept collaborating with our group for the theoretical contributions. And other people had worked on sort of similar models of cell migration in a variety of forms, and it was initially more of a challenge of choosing the right tools for the job, for the problems we wanted to study, which was how collective cell migration enables groups of cells in the developing embryo to cover large distances and find their targets so that the growth can occur normally.
Right. So maybe this is a good opportunity to introduce the problem itself, maybe a bit more formally. You mentioned already, like embryo development, and there are other applications, but in general, like what do we understand by collective cell migration?
Yeah, that's a good question. And it's worth clarifying. What I worked on mostly were groups of around hundreds of cells that moved together as a group. And the number of hundreds of cells is important because it means that it might be very hard to predict from just having an idea of what an individual cell does, what the whole group does. That's why the computation is useful, and the simulation is a good tool to give those predictions from the hypotheses. And the term collective is again important because you really need there to be some sort of emergent behavior. So when you put all the cells together and they migrate as a group, what you really mean is that the behavior is somewhat different from just each of the cells moving next to each other but without interaction. So it's the study of how the interaction between many cells gives rise to interesting things that would be hard to predict without the use of mathematics and simulation.
So you draw the boundary there that you can have just many cells moving maybe in the same direction and the same picture, but only now they're moving because they're interacting, right? You make the distinction, but like observationally, it may look the same or not?
Observationally, it may well look the same, and I think that's why you need models to implement your hypotheses and then challenge these models to come up with predictions to test your hypotheses. So maybe to give you one more concrete example, one of the problems we studied is that of neural crest migration in the embryo. And without going too much detail into the biological background, these are sort of multipotent cells that migrate through various places in the embryo to various distal locations, and they need to sort of get to the right places for the growth of the embryo to continue without further difficulties or without developmental defects. And one of the things we studied is whether maybe different cells within this group of a few hundred cells, a particular subset of these neural crest cells, whether some of these cells adopt different roles or different behavior in their migration. So rather than every cell just moving by itself and happen to be in a group of a few hundred other cells, we investigated the hypothesis, what if sort of some cells find the way and read out the guiding signals in the tissue environment, whereas other cells sort of just go with the flow and go with the group. And that, from our studies seems to be the case, or at least seems to be a useful description of what is going on, and led to a couple of predictions we could then feed back to our experimental collaborators to test in vivo, because after all, at the end of the day, the experiment will have the last say of what is really going on.
Mm-hmm. And how would you test this? How do you design the experiment to test this?
So that's a good question. It really depends on the predictions you can draw from the model, but also the things that are possible to do experimentally. So, for example, if you have your simulation of what you think is sort of a functioning migration, you might say, okay, what if I now transplant some of these cells into a different location, a different position within the group? How would that affect what we're going to see? Or how is it going to change if I take out a certain subset of the cells by sort of removing them or killing them? And one of these things is much easier to do experimentally. Transplantation, at least in the chick embryo, is sort of an established or more established embryology technique, whereas taking out individual cells you could, for example, do with a laser, but that has a bunch of caveats coming with it, because you create debris and might cause signals to spill into the environment that other cells could read it. And that's not something you have an hypothesis about in the model, which is why that would be a test that's more difficult. But, at the end of the day, you want to create a perturbation in your simulation and try to match that experimentally, either surgically or chemically, or even just through observation with genetic markers or analysis of the distribution of cells with their morphology and see if you can match what your simulations are suggesting. And that requires a constant back and forth between the experimentalists and the computational biologists, which is one of the really enjoyable parts of working interdisciplinary, I think.
Yeah, interesting. I already see a sign of a multidisciplinary approach, because what we're talking about very much sounds like a physicist's approach to formulate a model and then think about an experiment that would falsify or confirm that model, that theory.
Yeah, that's a good way of putting it. I guess with my background being in physics, what I usually strive towards is a minimal description, implementing the hypothesis that we have as a team of researchers, and to try to reproduce the biological observation in question.
Yeah, and a particular hypothesis that you scrutinize in your recent paper is the heterogeneity, right? So can you explain what that hypothesis assumes? Yeah,
so maybe let me go back a bit to say where that motivation came from, if you'll indulge me. So in most of my PhD work, we looked at this neural crest migration and looked at a form of heterogeneity with different cells adopting different navigational states or different migratory states. And we think that, as I said, that was a useful description. And, there was quite a bit of evidence in multiple papers to support that. Now, when you talk to other researchers, there's always, I perceive there to be this dichotomy between some researchers who wanted to characterize everything there was to characterize about the system on the one hand, and sort of other researchers championing parsimony to an sometimes absurd degree on the other hand. And the extremes would be that if I measure every transcript in every cell, I could say every cell looks different and looks unique. Whereas I could, on the other extreme, ignore all the detail I see under the microscope and say, no, every cell must be identical, which, at the embryo level, at least, we know is not true, or is not necessarily a useful description. So motivated by my experience in the PhD, I then sort of thought, how can we, think about measuring heterogeneity in cell migration? There are many different ways that one might measure this, but essentially wanting to say that if we use models to compare what we'd expect to see under a given hypothesis, we can be much more confident about whether some of the heterogeneity we measure is something biologically relevant or is simply sort of an artifact due to our limited measuring capability. So maybe to be a bit more concrete, what we looked at is originally whether some cells move ahead of others and sort of lead the way, and we established that at least in some of the neural crest cells in the chick embryo to be a useful description. But a more general way of how you might measure this is by tracking cells and then measuring which cells move before others. And you can do that using a technique called delayed correlation analysis. So if you look at how the movement of two cells correlates, if one cell always moves ahead of another, you would have the highest correlation between these two cells with a certain delay. And by looking at how large this delay is and whether it's positive or negative, you can establish how much heterogeneity of movement there is, so how much leader-follower heterogeneity, if you will, there is. And that's really a technique that came from the analysis of collective movement of animals. So people have studied these in migratory birds, where, of course, you can have long observation times by essentially tracking birds in GPS. And you can also have repeatability of the experiment while maintaining the identity of the birds. So if you collect the birds and keep their tags, you can do the experiment many times and measure every time which ones move first or not. But if you study cells, while this measurement technique might be an attractive statistical tool you can apply to your cell tracking data, you're really much more limited because you don't have very long observation times, especially if you culture the cells or if you look at them in the growing embryo, there will be limited amount of time that you can observe these cells. And secondly, you can't really repeat this experiment very well, maintaining the identity of the cells, because once an experiment in cell culture or in vivo in the embryo, once an observational period is finished, it's often the case that, the cells are done for, if you know what I mean.
But that cannot be the definition of heterogeneity, because I think in your paper, right, you say that the collective movement is possible without the heterogeneity. So you cannot define heterogeneity as just this leader-follower pattern, right? You mean something else by that.
Right. So that is just one example of heterogeneity. And you're right. I pointed out in the open biology perspective that, we have many models of collective movement that are working perfectly well without any heterogeneity, right? So I can, and people have done this, made simple models of flocking particles that sort of loosely resemble flocks of birds that just have every agent, every simulated bird or every simulated cell be identical, and you get a group movement that is collective in the sense we described earlier. Now, in nature, that may be different, and therefore it's important to try to, first of all, measure heterogeneity, but then also compare this to a suitable null model. And what we did essentially in the cell systems article is to say, here's an example of heterogeneity that you can measure. And in our case, this was this leader-follower heterogeneity in the form of delay correlation analysis. And when I measure the same property in my simulated migration of identical cells, so a group of cells without any heterogeneity, I still get a non-zero measurement. And to a statistician, that might be obvious because there'll be some correlation just due to chance in small sample sizes. But I thought it was really a point worth communicating to biologists because I saw the appeal of these statistical measurements and saw them being used in the literature on real cells. But thought, there's a cautionary tale to be told here.
Okay, so when you sort of refute this heterogeneity, you're not saying that there is no heterogeneity, and you're referring to like heterogeneity in the cells themselves, not in their roles, right? But you're saying that heterogeneity is not a causal factor, or maybe not the most important factor in collective movement. Is that correct?
That's partly correct. So you're right in pointing out that I'm not trying to say there is no heterogeneity. I'm saying that if we measure it and want to claim that there is heterogeneity, we have to be more careful because we really have to compare it to the models that represent our underlying hypotheses, right? So you might have some cells and you measure something about their migration, and then you might do a statistical analysis to say how the migration of some of the cells is different to that of other cells. And now the answer to whether that is to be expected or not, these differences really depends on your hypothesis of what the behavior of each cell is. So your model, if you will, right? So I can write down an equation of how I think that each cell behaves. And then if that is identical for every cell and still gives me the same expected or roughly the same expected difference in statistical properties of migration, then the measurement we have done is not sufficient to infer heterogeneity of the kind described in this population. But on the other hand, I'm also not making a general statement about whether heterogeneity is important or not in general in collective migration. We've seen examples where it is important, such as my work with the experimental collaborators in the neural crest migration, and I've shown you a sort of computational case study where it clearly isn't important, and we might get a false measurement. But whether it is or isn't really depends on the biological application.
Mm-hmm. And so one form of heterogeneity that I have in mind is the differences in gene expression in different cells, right? So has there been any attempts to measure it, or is it just hard to simultaneously measure gene expression and like the movement patterns?
Yeah, so that's a great example. The work I sort of illustrated just now is, of course, based on cell tracking data, but a very common and attractive form of heterogeneity to look at is of course the differences in gene expression, and that's something you might want to correlate with the differences in migratory behavior. And we've done this to some extent with our experimental collaborators in the neural crest, where we've isolated cells first in bulk and later individually to look at single cell gene expression of different positions within the group, and from that sort of identified, differences in heterogeneity that are consistent across different time points and unique to, say, a certain set of cells that are, in the case of the neural crest, located at the front, at the leading edge, which, by the way, is also something that our simulations were useful in guiding the experiments towards, to really focus on where and at what level of scale do you want to characterize heterogeneity. And from these profiles of a dozen or so genes that were consistently different in this group of cells that seem to be migrating differently, you can then either go and try to isolate markers that sort of maybe label this difference in cell state, or you can try to manipulate the cell state and perturb it by, like we did, identify transcription factors that sit upstream of this profile of gene expression at the leading edge of the cells. And when you then overexpress or knock down these transcription factors, you can again compare the outcome of the model to what you see experimentally. So in our case, this involves overexpressing this genetic profile that is characteristic of these cells in a leader state in the neural crest, which then led to a failure of migration, but a failure characteristic in that cells still migrated at as far as they would in the unperturbed group of cells in the embryo, but at a much lower density. And that's exactly what we saw in our simulation when we mimic this perturbation by increasing the proportion of cells in a leader state partway through the simulation, like you would in the experimental perturbation.
Wow, that's amazing, the fact that you can do something like that. Let's go to your model. How does it work? If someone wanted to make a similar model, what would they have to do?
So the first thing you really have to do is either know the biology or talk a lot to the people who do know the biology, because that'll inform you what details are important to include in your model. In my case, I've mostly worked with what you call agent-based models or individual-based models. And that just means that you represent each cell as its own object in a simulation. And the reason why we do this is because we only look at a few dozens or hundreds of cells. So the stochasticity in the individual behavior can still be important. And secondly, because the heterogeneities that we looked at were also on the scale of, maybe a few or tens of cells being different. If you had much higher numbers of cells, you might consider different mathematical models, such as continuous partial differential equations, for example. But in our case, really, there are these individual-based models in which you have fairly simple and not too computationally intensive simulations that implement your hypotheses as equations or sets of rules that each of these simulated cells follow. And if you want to make a start on these kind of things, you can even just go to our most recent paper and download the code, which I think is on GitHub.
Oh, cool. I didn't see the code. So if you send me the link, I'll include it in the show notes. And this model that you used, I believe it was borrowed from this like BERT models, right? So maybe you could explain in more detail the model and like the interactions that you include there.
Yeah. So in this most recent article in Cell Systems, I really tried to, I practiced the opposite of what I just preached, in that I tried to pick a very generic model. And the point of that was to show that this cautionary tale we were telling about the heterogeneity you might measure potentially not being real, I wanted that be not specific to the particular model details that you could, of course, argue with. So for this, I really tried to have a fairly general model of a group of cells moving together. So we stuck to what's called a self-propelled particle model, and that's just saying that each particle or each cell in the simulation moves out of its own action. And these, there's a long history of these models in at least phenomenologically describing bird flocks or insect swarms or fish schools. But they've, even sort of 15 years ago been adapted to be a bit more realistic for cell migration. And there's really just a very few ingredients in this model. And essentially what is important is that I told you that each cell moves out of its own action, and the interactions with other cells come in the form that the direction of a cell at a given time point is given by a combination of three different terms. And these three different terms are, first of all, some alignment with other cells. So this would be cells nearby move in a certain direction, and therefore you will tend to move in a similar direction in our case. Then secondly, there's some intercellular forces, so some attraction or repulsion. So in addition to aligning, you might want to stay close to other cells because you might have some adhesive forces, but at the same time you don't want to be too close because there's some volume exclusion, and if cells become too squished together, there might be a repulsive force. And then thirdly, the last term influencing the direction of movement would be noise, and that could be just to implement some intrinsic variability in the cell's actions, or it can also be, as it was in our case, to represent the fact that the interaction with other cells is noisy. So if there's some sensing involved of sensing the position or movement or chemical signals of other cells, there'll be some noise associated with that. And we've represented that again just very generically as a noise term on the direction of movement. And these rules, by the way, I should say, really date back in the conceptual nature to things like the Boyd model, which I forget when they're from the 70s or 80s even, which were used to describe just do graphically realistic simulations of bird flocks. Of course, we have working with much later iterations of these models that are more appropriate for cell migration. But really, at the end of the day, it's just that the interactions are alignment, attraction, repulsion, and noise.
Mm-hmm. And so then you ran the simulations with this model, and what did you find?
Yeah, so first what we found is that just by varying two of these interaction parameters, you really get a great range of morphologies of cell migration. So this was to make the case that this is really a suitable model to describe a range of settings that you might see in collective cell migration. So we found, for example, if the noise is relatively strong compared to the other two interaction terms, you just get sort of these disordered clusters that more or less look like they're diffusing around. Whereas if you have high intercellular forces, you get these static clusters where cells stay in the same position relative to their neighbors, and that is maybe a bit more like a solid tissue. And if you had very low attraction repulsion, you got these disordered groups that sort of break apart because they don't stick together sufficiently and move in different directions. And then, of course, most interestingly, at intermediate values of alignment and attraction repulsion, you got these moving clusters or streaming groups of cells that really look like a little a swarm or like a little crawling colony. And then we went on to look at this range of different simulations and do the analysis of heterogeneity in these different settings. And what we found there is that it's fairly easy to get non-zero heterogeneity in these simulations, which I should remind you, in these simulations we explicitly set that each cell is identical, and all the variability that comes in there is just to random initial conditions and intrinsic noise that is causing some chance correlations between cells. So you could easily find that certain settings of the simulated cells look like they're more or less heterogeneous than others. And even systematically so that with certain parameters such as the coupling between cells, their attraction repulsion, when this was stronger, it consistently looked like this measurement of heterogeneity was bigger, even though, as I said, every cell in this first set of simulations was exactly identical.
Right, and this is a bit confusing to me because I think here you're using maybe a different definition of heterogeneity that you gave in the beginning, right? And like when you say you measure heterogeneity, if we assume that heterogeneity is something inherent to the cells, right? You can just assume that they're heterogeneous or homogeneous, and then ask whether that heterogeneity affects the results, or maybe I'm misunderstanding this.
Right. So what we tried to do in this article was to say, if you measure heterogeneity based on cell tracking data, whether you can reliably measure it, or whether you can get measurements such as you would find, for example, in the literature, based even from models of homogeneous cell populations. So what we did first is start out with the simulation of a homogeneous cell population, then measure the heterogeneity in the resulting cell tracks, the simulated cell tracks, using this delay correlation analysis, which I described earlier, which essentially just tells you how often does a cell move first as opposed to later compared to other cells. And that sort of constitutes a population measure of heterogeneity. It gives you a distribution of delay times between cells. And the wider this distribution is, the more you would say some cells are moving first and some cells are moving last. Right? This is exactly what people have done in birds and also in mammalian cell cohorts so far. And what we said at first is that you can get these situations that look like some are moving first just based on homogeneous populations of cells and limited observation times, as you would find biologically. Then you can, as you said, you can do another thing, which is to assume heterogeneity and then ask, how does this affect the migration, the collective migration? And here again, you have to consider how you would measure this effect. And in this most recent study, we repeated the same analysis, but now on a group of cells where we, in the simulation, explicitly put in a subset of cells that behave differently by having a preferred direction. So this would be the example of, a few cells following directional signals, and the rest of the cells just interacting with the group. And then again, you can do this delay correlation analysis, which I'll remind you is just one possible form of heterogeneity in the migration that you can measure. And we found that in the populations with implicit heterogeneity built in those simulations, you still get similar amounts of heterogeneity measured. The systematic biases are different. So if you vary the parameters, that will change the heterogeneity in different ways. But importantly, this is connecting back to the experiment. If you look at experimental data that is out there, you could equally well describe this using models of homogeneous cell populations and models of heterogeneous cell populations. You would just have different resulting parameters in your model, if that makes sense.
Mm-hmm. And you draw attention to like the measurements being done over a short period of time. So what happens in your model, in your simulation, if you observe them over a longer period? Does this leader-follower pattern, does it break down?
So you're right that obviously I could run my simulations for much longer, and indeed we've run them for longer, than restricted analysis to a subset of these simulation times. If you had an infinite amount of simulation time, and if the system is suitably stochastic, you would expect over long times these correlations just to average out. So if some simulated cells move first, they should move last just as often later on in the simulation, for example. So yes, you would expect that with infinite observation times, these measures of heterogeneity in the delay correlation analysis would eventually dwindle down. And so you could even try to then, use models such as this to design your experiments and design your experimental observation times to see, how long would I really have to, how many data points in my cell tracking would I have to take to reliably be able to distinguish between, homogeneous behavior, migratory behavior of these cells or some intrinsic heterogeneity.
Right. So are you saying that like the theory predicts that, but in your simulation you didn't run them, even though you ran them for a long time, you didn't run them enough to observe that?
So what we did is to use our simulations in a way that would be representative of the experiments that have been done. So there would have been fairly little point in showing you a simulation of homogeneous cells and then showing that, with infinite simulation times, I can indeed show that they are homogeneous. The point we were trying to make was much more to say, if you have, as many or even quite a few more data as you would do in typical cell tracking experiments, then that is not necessarily enough to distinguish between the heterogeneity being real or artifactual. But of course, that depends on the model.
Right, but I think the point would be exactly what you said, to see whether, during a realistic time, maybe a bit longer than was done in real experiments, right? But there's a difference between, I don't know what the typical time scale is, but let's say we observe them during 1 minute, and there's a difference between saying, if we only observe them for 10 more minutes, we would see the pattern reverse. Or if we would observe them for another 100 years, we would see the pattern reverse.
Yes, you make an excellent point, and I understand what you were trying to ask better now. So I'd have to check my numbers, but we probably had an order of magnitude more data points in our analysis of the simulated cell tracks compared to what has been done in the literature on cell tracking. And of course, in the original example that I mentioned, where similar analysis have been done to flocks of birds, that's a different story, because there you have higher temporal resolution and longer observational time. So I don't know how many data points they would have had there. So I guess what we're saying is, even if you have an order of magnitude more data points, that may not be enough. Now, that depends both on your observational time and your sampling frequency. And I also should also stress that it doesn't necessarily doom any attempts to measure heterogeneity in cell tracking, because it would depend, again, on your underlying model. But in this generic framework that we've adopted here to be suitable for a wide range of applications, it was the case that even with an order of magnitude longer observation times, you can still see signatures or semblance of heterogeneity, as we called it in our title, in these simulated cell tracks.
Mm-hmm. And at the beginning, you said that a lot of researchers now approaching this cell migration problem from the computational perspective. So what other interesting models have been proposed? And maybe let's just restrict this to discrete models, not continuous models, but what are some alternative models to this one?
Sure. So I guess there's no strict alternatives here, in the sense of, we didn't have a particular application in mind. So, if you had an issue with the model, then it would be more with the, I guess, functional form of the equations, for example. But really, the debate needs to be had over the particular applications. So maybe if I just illustrate what kind of different models people have for different biological settings, there are, for example, models that include a much more detail at the subcellular scale in terms of, for example, the shape of the cells. So there's models that through a variety of formalisms, might even simulate the protrusions of cells and how that affects interaction. Or there might be a lot more realistic mechanics in some of these models. So, for example, in models of epithelia, so this sort of more two-dimensional tissues where every cell is densely packed and connected to other cells, you could represent sort of cells as little polygons with vertices, and then these vertices interact with each other with certain forces, and that would be a vertex model that would be appropriate for certain applications, but not others, for example. So really it depends on the dimensionality of your biological setting and the topology of your cell neighborhoods and the kind of interactions you're considering. Another important breed of models are the so-called hybrid models, where you combine the sort of discrete modeling of different cells that move with continuous representations of chemical signals that these cells might produce or interact with. And that's something we've done in our work in the past as well.
Mm-hmm. And these models, I assume they can be made both homogeneous or heterogeneous depending on how you parameterize each individual cell, right?
That's right.
In the paper, you proposed this model that you described earlier as a null model for the other model, which is the heterogeneity model. But this heterogeneity model hasn't ever been formalized.
No, so we do this in our paper. So the heterogeneity model in our case would be the same model but parameterized so that a subset of the cells in the group have different interactions, or as I said, have a preferred direction of movement to represent the fact that they might be following some chemical signals. And we do this in our paper to investigate, how does this affect the measurements of heterogeneity? Can we describe experimental data just as well?
Right. That makes perfect sense. What I was saying is, for every model, so for your model, right, you can add heterogeneity back there, right? And for, I presume, every other model, you can make them homogeneous or heterogeneous. But I got a sense from your paper that you were proposing your model as like sort of a generic null model against which it makes sense to check the data and other models. But what you're actually saying is, for whatever model you're considering, you should also derive the null version of this model where everything is homogeneous and see whether it still holds.
Yes, I guess that's what we're saying, is to say, for your biological application, choose an appropriate model, which might be similar to what we've done or might be quite different. If you're, for example, studying epithelia, you might choose a different model. And then if you're trying to characterize or measure heterogeneity, then compare it to the homogeneous model, right? And if you still see more heterogeneity in your data than you'd expect from your model representing the null hypothesis that every cell is identical, bar some intrinsic noise, then that can guide you towards refining your hypothesis to say, okay, I think there is heterogeneity, and then we can go back to a model, build it in, and again compare what we see in our measurements compared to the now modified model, and thus you get into the cycle of use the model to predict, test it experimentally, and then refine the model, which is sort of a common framework in quantitative biology or mathematical biology to do this iteratively many times.
Right. That makes sense. So in your model, the salient feature of your model is that the cells, they sort of align their velocities in maybe not in the common direction, but like in the direction of their neighbors. And is there a plausible, like physical or chemical mechanism by which this can happen?
Yeah, so that's a valid criticism. You might say that this interaction term that aligns the cell's movement direction might be considered unrealistic. We think it's okay because other people who've modeled the cell movement in more detail with sort of lamellipodia protrusions, these are just sort of extensions of the cell membrane in front of cells, have shown that sort of some short-range adhesive forces between cells that have a non-uniform shape can essentially be equivalent to an alignment term, just based on the fact that when cells maybe collide and have a certain shape and adhesion, the outcome can be such that they're effectively aligning their velocities. Another mechanism you could think of is to have some chemical signals that these cells secrete, for example, and that guides other cells to move towards them, and that would be more of an sort of attractive guidance mechanism between cells.
Yeah, but that only covers attraction, right? Because this cell cannot directly observe the velocity of another cell. So you can imagine a situation where, for example, I'm moving towards you, but at the same time you're moving in an orthogonal direction, but I cannot sense that until I come sufficiently close to you.
Right. So, yeah, I struggle to think of an example where you could sense that with the chemical signals I described. You're right. There might be, effects due to if you also actively degrade these signals by internalizing them. But there's other ways you could presumably sense this biologically, and I'm happy to be corrected by any listeners that have more biological expertise. But you could either, sense traction, for example, in the extracellular matrix or whatever medium you're moving through. You could even have modifications in the environment that cause you to follow the path of other cells, for example, and even in some of the neural crest studies that our collaborators from the Kalisa lab have done, you can see cells contacting each other with phallopodia, and then migrating in parallel afterwards, as opposed to towards each other. So there could be an effective alignment also through active movement and keeping your distance to a neighbor. That can have an aligning effect. If you come in towards another cell trajectory at an angle, and then you keep a certain distance to it, that will align you towards it as well.
Mm-hmm. Interesting. Okay.
But, you're right to say that if it's unrealistic in your particular biological application, then, you need to modify your model, and that might change what you expect to see.
Mm-hmm. But then we also have to explain not just why cells move together or in a common direction, but how they arrive at the right place, right? And I think just a homogeneous model without any external signals cannot really explain that, right? So you still need some maybe global chemotaxis or something.
Yes, you're completely right. If the question you're trying to answer is how cells get to the place that they move towards, then you need either some sort of confinement to keep the cells moving into a certain direction, or you need some gradients of chemical signals that impart a preferred direction on the movement of the cells. Or there's actually a third possibility, which is another thing we've studied in the case of the neural crest, which is to have a so-called cell-induced or self-generated gradient, which is to say that even if you have a uniform distribution of chemicals, if you have the cells degrade these chemicals by either producing other chemicals that degrade your guidance molecules, or by internalizing the guidance molecules when you sense them, therefore acting like a sponge that soaks up these guidance molecules, then you locally deplete the concentration of these guidance molecules, and that creates a gradient around you. And then if you're in a group, that imparts a direction to essentially move the group forwards or spread out at least. And there's some exciting work in many different biological systems that shows that may be the case in a variety of settings.
In your paper, you talk about, you compare two ways to define models. One is this rigorous mathematical way, and the other is sort of verbal, vague models. And you say that we should strive to bring verbal models into the mathematical land. But I'm curious, now that you have, let's say, this model, can you sort of do the reverse? Can you make a verbal picture of this mathematical phenomenon? For example, there are different patterns in which cells can move depending on the parameters. And can you explain how they emerge?
Yeah, that's a good challenge we should always pose ourselves to communicate our results. With any sort of truly collective effects or emergent phenomena, that's always going to be difficult, because almost by definition, it's hard to reason why something happens when 100 or more things interact in a certain way from just the description of how the individuals behave. You can do it at a coarse level, where you can say, in this case, when the attraction between cells is high, but the alignment of the directions is low, that'll give you some sort of statically ordered cluster. Whereas if you have some intermediate attraction, but very high alignment between cells, that will give you sort of, a stream of cells all moving in the same direction, but with only sort of loose positional order between them, for example. And just to touch back on what you said, the reason why we say the converse is true it's important to bring the verbal models in the mathematical land, as you said it. That's of course because the mathematics will tie you to the logic and will enable you to make predictions from your hypotheses when it becomes difficult to do so verbally, right? Because you might say, okay, so this cell does this, and then the neighboring cell moves in such a way, and then in turn the cells around it will move in yet another way, and by the time you get to, dozens of cells over many time steps, it just becomes infeasible.
Right. And yeah, I recommend all the listeners to check out your papers because there are many great points such as this one in there. Really enjoyed reading them.
Thanks.
Before we part here, I wanted to talk to you about multidisciplinarity. I imagine there can be two ways to have multidisciplinary research. One is for people like you who are well familiar with both biology and physics and mathematics to just carry it out. And the other way is to join multidisciplinary team where we have a physicist and a mathematician and a programmer and a biologist. So which one of these two do you prefer or advocate? Or
yeah, that's a great question. How to do multidisciplinary work and whether to be, an expert in a team of experts or whether to be a jack of all trades. That's difficult, because, if you bring something to the table with an expertise in programming or physics or a particular biological experimental setup, that can often be more efficient because, you don't have to learn everything from scratch. On the other hand, it will take a lot of communication time between the different experts to make sure they're talking about the same thing and talking at the same level. On the other hand, being someone who does both, for example, the experiments and the simulations, is very attractive because you will really understand every part of it, as well as you can, but it may slow down your progress. One of the reasons why I've opted to be a computational and theoretical biologist working closely with experimentalists is because I like the variety of different applications. And I got the impression that whenever I tried doing experiments, which I've done a bunch of times, it seems like you invest much more heavily in a particular experimental system, at least in developmental biology, which I mostly looked at in my PhD. You might become an expert in doing experiments in zebrafish or xenopus or chick embryos or cell cultures. And you may be able to do two of these or, change when you go from PhD to postdoc or from postdoc to PI, but it seemed much more of a commitment. And I guess I was feeling non-committal. But if you find something that truly fascinates you and you're happy to, stick with that for a long time, then I would recommend to go for it and do both experiments and theory if you can.
Yeah, and I was thinking there's probably a third way, which is maybe the most efficient, is when you have the specialists, but each specialist, like if it's a biologist, but she also knows a bit of programming and understands how to talk to programmers. And if it's a programmer, then he understands a bit of biology and understands how to talk to biologists.
Yes, that's a good point. And that's probably closer to what I try to do. So when I work with biologists, I try to, always at least observe the experiment once or maybe even do it if they let me to know what's involved, to make sure I understand how difficult it is and what the caveats are. And at the same time, it's very useful for the biologists to know enough of the mathematics and the programming so that they can understand what they're being told and also critically assess whether it makes sense to them. Trust is good, but understanding is even better. That's put it that way.
And do you have any advice for maybe a mathematician or a physicist who's listening to this and who would like to get into multidisciplinary research or biology in particular?
That's a good question.
Where do they start?
It depends a bit on the career stage. If you're looking to do, say, a PhD, I would look for some interdisciplinary training programs where maybe you can learn a bit more biology if you come from the theoretical side, or you can learn a bit more mathematical computational skills if you come from the biology side. if you're already a trained scientist in the sense that you've done your PhD or maybe a postdoc and want to move into that, you just gotta, start talking to biologists, maybe go to some of their meetings, to find out what fascinates you, but also where you think you can make a difference with your skills. You might see them talking about a problem that you think you can solve using your, statistical analysis or your simulation or your mathematical techniques. So really it's about communication and, finding a problem that you can really sink your teeth into.
Excellent. Well, Linus, thanks a lot for taking the time to come on the podcast. That was very interesting.
Thank you very much for having me.